First speaker of this session is Matt Kaeberlein. He's from University of Washington, and he's going to tell us something about, I think, AI and large screens and how we can find the drug against aging.
All right. Okay, thanks. So first of all, I want to thank Morten and Daniela and Alex for the opportunity to talk to you today. And I thought, given the theme of this meeting, I would talk about something different than I usually talk about. Some efforts that we're making in my lab to develop some tools to what I hope will dramatically increase the capacity for screening for new longevity interventions. So what I want to do today is hopefully convince you why that's something that's important and would be good to be able to do. And then secondly, tell you about the technology that we're developing to do that in C. elegans. And so this is a system that we have called the WormBot. It's a robotic system for lifespan and health span analysis in C. elegans that we have now coupled with neural networks to really automate the entire system. And it's really set it and forget it kind of system. You set the experiments up, come back in 3 weeks, get your data. Or at least that's what we're going for. And I feel like we are essentially ready for prime time now, which is part of the reason why I'm bringing it out to the community to get some feedback. This is really the work of several people in my lab, but primarily these two individuals. Jason Pitt, who's a senior research scientist in the lab, has developed all of the and Ben Blue, who's a graduate student in the lab, has really led the AI neural network development that I'm going to tell you about. So before I tell you about the system, I just want to take a step back and talk about why I think we really need the capacity to do intervention discovery at a scale that hasn't been possible before. And I think it's fair to say that at least when it comes to interventions, in many ways the field has stagnated. I think that we have been extremely successful in the last 20 years at discovering mechanisms, at starting to develop efforts to translate those mechanisms. But when you look at the actual magnitude of effect of interventions, it's really not that impressive. And this is just a list of probably the most common interventions that people think about, non-genetic interventions in laboratory animals today. And what's indicated here is that the largest magnitude of effect for lifespan is an intervention that was first published 80 years ago, right? Almost 90 years ago. And actually the longest lifespan extension from a non-genetic intervention that I know of is this experiment from Rick Weindruck and Roy Walford that was done in the 1980s, and that's the 65% caloric restriction. And then when you look at the pharmacological interventions that we're studying, by and large today, they're all substantially smaller. So rapamycin, of course, is the most robust and reproducible pharmacological intervention for targeting aging that we have today. It's my personal favorite. We're testing rapamycin in pet dogs right now in a clinical trial, but its efficacy is not as great as caloric restriction. It's not even close. And then when you look at the other interventions, they're not as effective as rapamycin, at least for lifespan. So I feel like we need to get beyond this, right? There would be, it's important for the field not only to find new interventions, but to find particularly effective interventions. And so I think we can ask, why aren't we finding better interventions? There are probably lots of reasons, but I think one reason is that we just don't have any great tools for screening for interventions at scale. We don't have particularly useful high throughput platforms right now. That means that we're not able to really do combinatorial screening, when I think that's actually where a lot of the bang for the buck is going to come from, is by combining interventions where you can get additive or synergistic effects. And I think also, we're largely stuck studying what we know already. There was a period where people were doing unbiased genetic screens and finding lots of new hits and generating hypotheses about pathways. But largely now we're studying the same 8 or 9 different factors. And so I think there is still a lot to be discovered outside of looking under the lamppost, and that's kind of what I'm trying to advocate for, is that's something we should be trying to do as a field, is look outside of the light and figure out what else is out there in the intervention space. So the system we've developed, I mentioned, is called the WormBot. We actually published a paper a couple years ago on the open source version of this system. Our first version was built with a commercial make block kit. So it was something that you could buy on Amazon at the time and build. I'm going to tell you a little bit about the design, and then I'll show you the newer version that we've got, which is substantially more robust. So the way this is laid out is the platform here holds 12 12-well plates. In principle, it can hold as many as you want. That's just the size that we made it. And in each well is an individual experiment. So there's 20 to 30 individual animals in each well. And what the robot does is it moves a webcam over each well, takes a picture, goes on to the next well, takes a picture, does that for all 144 wells, and then goes back to the beginning. So what you get is, over a period of a few weeks, a time-lapse image of every animal throughout their entire life every 6 to 10 minutes. And then we also take about a 1.5-minute movie each day of each well. And then we have some software that we developed to control the robot, so you can say what experiments are there and which wells should it look at and things like that. We've exported this version 1.0 to a few different labs who are using it, and that was great, gave us some beta testing to improve the system. And so now we're on our 3rd generation, and this, I think, is ready for prime time. No question we could make it better. We're not robotic engineers. We're not computer scientists, but it works really well for what we want it to do. So this version 3.0 is all custom-sourced parts. The motors are much more robust. You can kind of see Jason felt that it was important to put this emergency stop button here, I guess in case somebody got their head stuck in there. But it's pretty powerful, actually, the force that the motors can generate. And like I said, it's working really well. You can see it moving the camera and taking pictures. And I think the real breakthrough has come in coupling this with AI. So Ben has been working using a package called YOLO, which stands for You Only Look Once. It's a image recognition package for AI. And he has developed a neural network, which you can see it functioning here. So what you're watching is a movie of these time-lapse images, 6 minutes apart throughout the entire lifespan of these worms. And the neural network in every image is recognizing the worms, putting a blue bounding box around it. And then when the software has determined that the worms haven't moved for long enough and don't start moving later on, it calls them dead, and that's when it switches to yellow. And so, like I said, it works really well. And in fact, it works as well as a person as far as we can tell. So this is a comparison of a human being scoring the same data as the neural network by sort of manually going through these time-lapse images, and they're right on top of each other. It turns out, at least this is a little preliminary, but it looks like it also works pretty well for data off of the automated lifespan machine, which for any of you who are C. elegans aficionados, that's the flatbed scanner system that was developed a few years ago. It seems to work pretty well analyzing those images as well, so it might be useful beyond just data collected off of the WormBot. So we've been doing experiments, as you can imagine, as we've been going along developing this. I'm just going to show you a couple of hints of what we think that this system can do. So this is a data collection of about 381 experiments. Remember, each experiment is one well. It's about 20 to 30 animals. So this corresponds to almost 14,000 animals. This is from a collaboration with Sue-In Lee, who's in the computer science department at the University of Washington, where they took human data, a variety of different types of human data related to Alzheimer's disease samples, did interpretable machine learning on that data to predict genes that might be involved in Alzheimer's disease. We identified the worm orthologs, and then we tested those by RNAi knockdown in a late onset Alzheimer's disease model in C. elegans. And so each dot here represents the average of multiple experiments for a different condition, and each row is a different RNAi clone. And for the purposes of this talk, it doesn't really matter what the specific RNAi clones are. What I want to show you is that the empty vector, which is these bluish dots here, and the treatment, which is the dark blue dots, for each RNAi clone, you can now start to get a real feel for what the variation is from treatment to treatment, okay? And remember, these are now, each dot is an average of multiple experiments for that treatment. And in most cases, there's no effect. The red dots, which are all the way to the right, are DAF2. So those are living longer. That's our positive control. And again, you can see there's variation from a treatment to treatment, but by and large, the DAF2 is always to the right of the empty vector, meaning that the positive control worked. And then the other two colors, the orange and the green, are actually early hits, both electron transport chain genes, which is interesting biology, that we started including as positive controls. And again, you can see that while there's variation from treatment to treatment, these things are almost always to the right of the empty vector. Again, consistently giving positive effects, which is what we would expect for our positive control. So you can also look at this now as individual experiments where it gets noisier, right? But one of the powers of this is you can do the experiments as many times as you want. You can do 12 replicates if you want and get a feel for what's the distribution of the controls and what's the distribution of the treatments and how often do those tails overlap. So it's nice from that perspective. We've also been doing some drug screening. So this is very early. This is part of Ben's thesis project where he's looking at drugs that have been reported to extend lifespan in C. elegans on DrugAge, which is probably the best database for drugs that have been reported to affect aging. This is a collection of about 200 experiments, about 4,500 animals. Each row here is a drug. Now this is showing you the percent lifespan extension, and it's probably a little bit hard to see, but the 0 is right there in the middle. So most of the drugs aren't having a big effect. This is, again, a distribution now of individual experiments. But some are. Some are far to the right and having big effects on lifespan. And doesn't really matter what the specific drugs are, but if you're interested, these are the ones that look, at least for now, like they're having a big effect. So this is preliminary, but again, I think it illustrates the sort of magnitude of what you can do. Our goal is really to test most of the drugs that have been previously reported to extend lifespan in C. elegans and see what we see. We've also been incorporating fluorescence microscopy, and I think this is going to be pretty powerful. So you can imagine lots of things that you could do if you could get time-lapse imaging of fluorescent proteins in worms over their entire lifespan. In this particular case, this is a polyglutamine YFP animal. Again, I'm not going to talk about anything interesting that's happening, except that you'll notice these red circles pop up. One thing we've noticed is that for polyglutamine and for other fluorescent proteins, at end of life, worms show this what we call fade to black phenotype. Now, that's not super surprising, right? After a worm dies, of course, eventually the fluorescence is going to go away. But it happens pretty quickly. And one of the interesting things is it doesn't happen the same way in every animal when they die. So we're wondering if heterogeneity in the fade to black might actually tell us something about heterogeneity in modes of death. So we're looking further at that. I think there are a lot of applications, though, that you could imagine with coupling fluorescence microscopy to time-lapse imaging. So what we're doing right now, we have about 4 WormBots in the lab. We've retired our first and second generation WormBots. Really, our bottleneck is temperature controlled. And for anybody who works with C. elegans, you know why being able to control temperature is really important. So we have 2 WormBots that are temperature controlled at this point. We've dedicated one of them now to doing this sort of pilot drug screening with an FDA-approved library. This is just getting started. But we're testing each drug alone and in combination with metformin. And this gets back to that combinatorial interaction that I think is going to be really important. So metformin works really well for us in our lab. And so I think hopefully what we'll find are things that synergize with metformin, and probably we'll find things that block the metformin effect. And I think that doing this for 5 or 6 of the most prescribed drugs in people might be really interesting and informative. We are doing this both in wild-type animals, which are N2, and in an early onset Alzheimer's model where they get paralyzed in about 3 days, so that'll be much faster throughput. We're currently also optimizing our ability to quantify activity. So I showed you the neural network could identify where the worms are. We are working to be able to track the same animal from image to image over its entire life. We can do that for the second half of the experiment pretty well already. It gets harder, obviously, when they're moving around more. But if we can follow individual animals, we can actually get a pretty decent estimate of total activity over the entire lifespan. We're developing methods to look more closely at morphology and then testing higher resolution optics, which certainly should help with that. So where do I hope that we'll be able to go? I picked 100,000 because it's a big, nice number. Nothing magic about 100,000, but I also think it's pretty easy for me to envision how we get there from where we're at now. And I think if we can get to 100,000, that really opens the door to starting to be able to do unbiased drug screening, looking at maybe genetic models of human disease in C. elegans and repurposing drugs. And then, as I've already said a couple times, what I'm particularly interested in is combinatorial drug screening. And we're not limited to looking at the combinations that we think are most interesting, like rapamycin plus metformin. We can really expand what we want to look at. So how do we get there? So one WormBot can do about 2,500 experiments a year. That's at 25 degrees. That's assuming 3 weeks per experiment. Obviously, if you worked in different models, it might be shorter or longer than that. But let's just say 2,500 a year. We've got 4 of these in my lab right now. Really what we're limited by is space and temperature-controlled space. But it's working. It's robust. I see absolutely no bottlenecks to expanding this by tenfold or even more. And if we can do that, that gets us to 100,000 interventions a year. And really that's, I think, a low bar. Like I think we could, as a field, certainly get far beyond that. So I'm hopeful that we can actually expand this system and get to the point where we can really do sort of massive scale intervention testing in C. elegans. Obviously, it would be great to be able to do this in a mouse. I think it's going to be a while before we can get to the point where we can do intervention testing at a scale like this in a mouse, but we can get leads that can then be tested in mice. So I'll just finish up with a few final thoughts. So one thing I think it's important to appreciate, so I'm a big fan, by the way, of all the applications of AI that are being used in aging research right now. But I think it's important to appreciate that most of them are built on what we already know, right? They're built on what we've seen under the lamppost. And I think this is particularly important for the methods that are trying to identify signatures associated with interventions that you then might be able to test in cells, for example. Those signatures are based on interventions that we already know about. So my concern is that if we use those tools exclusively, we're going to miss everything else that we don't know about yet. And that's why I want to advocate not to stop doing lifespan experiments. I think that that's kind of the foundation for where we start. That's the gold standard. I also want to make the point, we've only explored a tiny fraction of the intervention space to this point, even among genetic interventions, right? All the RNAi screens or deletion set screens that have been done are loss of function. Nobody's done a systematic gain of function, for example, or random mutagenesis for lifespan. If you look at drugs, it's an even smaller fraction of the intervention space. So DrugAge lists about 970 records for C. elegans. That's about 400 molecules, right? That's a tiny fraction of all the individual drugs that could be screened, let alone all of the combinations that could be screened. So we really have a lot, I think, to discover out there. And my final thought is I think we need tools like this. Again, I feel like we're probably missing some really important biology if we don't start looking outside the lamppost. And so I really want to advocate for that. And hopefully we can expand our view of aging biology in this way. And so with that, I will stop. And if I have time, I'll be happy to take any questions or comments people might have.
Really great presentation, Matt. I have a question. I'll also check the Slack, but I have a question that I asked Andy Dillon about 10 years ago or something like that when I was this tall. And
and my beard was still black back then.
Yeah, maybe. Yeah. No. And I asked him what actually kills a worm. What did they die of?
So I think there are differing thoughts on that. But my view is that at 25 degrees at least, proteostasis or collapse of proteostasis is a major driver. But I don't think we can explain everything that's been reported to extend lifespan as purely affecting collapse and proteostasis. So, but I think the real answer is we don't completely know, and maybe this fade to black phenotype will tell us something about that.
Very interesting. Do we have a question down here?
Yeah, great talk. Very interesting. Just one question I came to my mind when we think about the talk of Maria Ermoleva earlier this day. Can you envision to use the same system also for late life intervention screens? Because I think what is really what we may really miss is, with these lifelong screens, you affect things early in life that have this lifespan effect that may not work at old age. But when it goes to translation, maybe old age is the most critical age when the intervention has to work.
Yeah, it's a great question. So the easy answer is, yeah, it's trivial, right, to do a late life intervention on this system because you can take the plates off and on the robot whenever you want to. You can just pause it, right? So it would be easy to imagine, aging the worms out on the device till whatever age you think is appropriate and then adding your compound or whatever your intervention is. So yeah, that's trivial. The other point that reminds me I want to make is I think, and I'm concerned about this as well, we don't want to put all our eggs in the C. elegans basket. So are there ways to develop a system like this for other model organisms that are amenable to those kinds of approaches? And we're thinking about that, and we're thinking about some other options for that as well. So.
I would recommend looking at Michael Peters' poster. We're doing the same with Drosophila. So,
yeah, great. So
that's a good thing. Anyway, sorry for that, for pitching that just out of the blue. I apologize. There's an upvoted question here from Slack, from Dmitry Kryukov. Brilliant idea to automate experiment. Is it possible to implement reinforcement learning algorithms to conduct more effective series of experiments?
So if I understand the question correctly, I think what's being suggested is that you could actually use something like AI to then, once you've got a large enough data set, predict what other things are going to work. And absolutely that's possible. I mean, people are doing that already today in a variety of contexts. And I like those approaches, but as I said, my concern is as soon as you do that, you're sort of giving up on what you don't know already. Because if you're training the system on what you know, and there's a whole lot of unexplored space, there's no guarantee that what that system is going to pick as hits going forward are going to reflect what might work even better in the unexplored space. So the answer is yes, but my view is I'm not smart enough to know when we get to the point where we know enough. So I'd rather just do it as unbiased as possible, right, if we can do it that way.
Yeah, that's a great point. And I think we'll stop here and let's give Matt another round of applause. Fantastic talk.
Thanks.